Hello, everyone. My name is Zhinan Yin. It is my great honor to introduce our recent publication published in Phenomics. The title is Immune Aging Evaluation of Peripheral T and NK lymphocyte subsets in Chinese healthy adults. As we know, aging process is primarily composed with three phases with different hallmarks. The first one we call the cause of damage. That's including seismic evolution, genetic alteration, loss of proteostasis, and mitochondrial dysfunction. And the second hallmarks we call the consequence of the damage. That's including repertoire reduction, naive memory imbalance, T cell synthesis, and the lack of plasticity. And the last stage, we call the integrated hallmarks. That's integrated of damages such as immune deficiency and inflammation. So aging indeed is a complex process, involves many physiological and biological changes. And T cells and NK cells play a very important role in the development of age-related immune repairment. So our experiment design is connecting up to 70,000 individual samples and analyze up to 36 functional markers. These marks are including differentiation states and exhaustion markers. Then we use this model to build an immune age prediction model. Here are the sample distribution. As you can see, we have more females than males. Most of the samples between age 40 to 60, and most of the samples collected from the south part of China. Only a small fraction come from the north part of China. So from our data, we first calculated the absolute value of slope for population size age linear regression of each immune cell type. For example, the highest is naive CD8 T cells, and followed with functional CD8 T cells, which is CD28 positive CD8 T cells, and then terminal differentiated CD8 T cells, and then exhausted NK cells, and the number of V delta 2 gamma delta T cells. As you can see, some of the parameters reduced with the age. Some parameters increased the value with the age. So we then use a random forest machine learning model. We use our immune parameters to predict your age. As you can see, the RC right now is 78.4%. Indeed, we use 5 different batch of the data. As you can see, the correction rate is more than 70%. So finally, we also use 2 cohorts. One is the healthy donors, and one is the cohorts with chronic diseases. We demonstrated that here, people with chronic diseases are predicted older than from healthy donors. And consistent with the slope data, we also calculated the important ranking for the immune cell type. These are very consistent with the slope data. As you can see, the most important 6 parameters that contribute to the age of our prediction. And finally, we also provided the percentage and the absolute number of T cells and NK cells with different age group. So this the data. So in summary, from 36 immune parameters out of 70,000 individuals, we applied machine learning model to develop an immune age model. Using this model, we identified the most influencing immune age factors and predicted immune age of individuals. To the best of our knowledge, our data set is the largest cohort of peripheral blood immune parameters and the first ever immune age prediction model. We also demonstrated that CD8 T cells, NK cells, and gamma delta T cells are the key features affected with immune age prediction. Human immune age might be different from chronological age. From our study, for the first time, we demonstrated that people with chronic diseases have older immune age than those cohort with healthy donors. For the future perspective, we should collect more samples from different regions of China, with their basic test results, including liver and kidney functions, and with more samples from ages 60 to 80 years old, to develop a more precise immune age prediction model. We call it version 2.0. Before closing my presentation, I greatly appreciate all the donors participate in the study. Thanks a lot to my colleagues and research teams. Without their support and help, this study would not be possible. Special thanks to Pure Biotech to handle all the samples and analyze using their standard protocol and quality control. That's all for my presentation. Thank you very much for your attention.